started. We're going to lecture now. Unless you guys want to talk about amino acids.
Are you in this class?
No. I like to learn about amino acids.
We'll probably talk about it in Chem 103 if you haven't already. I'm surprised Sean has done any biochem yet.
So we're kind of doing like geochemistry kind of stuff. So like silicates and
Okay.
all that kind of fun
You know,
stuff.
you just have to take it.
Yeah. Well, I will be in this class anyway, so.
All right.
From a year from now, I guess I'll be learning about amino acids.
Is that the throat, Jared?
Someday. They're cool. Yes. I have protein. Protein is
That's what we're made of.
50% of your bio weights. Yes.
We are amino acids.
So, that's true. Amino acids. So today we're going to talk about amino acids, and then we're going to talk on Thursday. Today's Tuesday, right?
Yes.
For some reason I feel like the schedule is. Am I off? Anyway, the next lecture will be about amino acid. What week is this? Oh, you know what? Yeah. Well, shucks. We were supposed to talk, carbohydrate react. I see. We already got into the carbohydrate reactions a little bit. We talked about carbohydrates. So we're going to move on. Everything's going to bump up a little bit. So that'll give us a little bit more time to talk about lipids. So yes, while this is not quite what the schedule says, we are still good. Today we're going to talk about amino acids. And then on Thursday, we'll talk about how we put amino acids together to make peptide bonds that make peptides. Peptides are just small fragments of proteins, okay? So amino acids are the building blocks of proteins. So proteins are the workhorses inside your cells. They're also what make up a large percentage of your body. And in fact, if you were to take all of your dry weight, meaning if we were to dehydrate you completely and turn you into dust, and then separate that dust out into nucleic acid, fat or lipids, carbohydrates, and proteins, 50%, give or take, would be proteins. All right. Proteins make up our hair and fingernails in the form of keratin. Proteins make up our muscles in the form of myosin and others. They make up our ligaments and our tendons, so our connective tissue, our articular cartilage, the stuff in between our joints. That's all collagen, that's a protein. Collagen is in our skin. As you get older, you make less and less collagen. You get wrinkles. You live up here in a desert. I guess it's not really a desert, but it feels like a desert. It's so dry up here. Your skin dries out and you get even more wrinkly, right? Amino acids outside of the biochemistry context are often in performance, athlete, weightlifting, things like that. You think of amino acids to build up proteins, right, to repair from a workout. And so today what we're going to talk about is what is an amino acid? What's the structure of an amino acid? What are the properties of amino acids? So, turns out they can be acidic, or they can be basic, or they can be neutral. They can be ions, or they can be protonated or deprotonated depending on the pH and the pKa values. And so we'll talk about that. And then we'll get into a few reactions on how to make amino acids. And that should wrap things up nicely for today. And then, like I said, on Thursday, we'll get into protein synthesis. We're going to do PCR today. So I think what I'll have us do is for the lab, we will do PCR with some test DNA samples or standards. And then if you want, we can extract DNA from our cheek cells and or hair follicles and see if we have these ALU repeats. Okay, I've never done that part. I've always just had students run the standard DNA and just put it through a gel and see it, but I think we'll have time if you want to see if you have this ALU repeat in a certain gene and whether or not you're homozygous negative for it, meaning you don't have any copies, or you're homozygous positive, meaning both parental alleles have that, or if you're heterozygous, one's there and one's not. You won't know which one you got it from, of course, but okay, cool. So here's the outline for today, and then here are the amino acids. Not the amino acids, but some amino acids in a generic form. And it's just drawing them out here to show that they fit together kind of like puzzle pieces in a way. It's a dehydration reaction. So you'll recognize this as a carboxylic acid group, right? And you'll recognize this as an amino group. That's where the word amino acid comes from, because every amino acid has a carboxylic acid group on one end and an amine group on the other end. And to then stitch these together, which we'll talk about more next time, to make peptides, you will essentially cleave off this hydroxyl group and cleave off one of the hydrogens, right? So H and OH go off to form water, and then we attach this amine group on to that carbon in place of it, okay? And then that forms the peptide bond, and then it turns out that these amino acids each have different characteristics, not based on that. That is the standard common denominator for all amino acids. What makes them different is the R group. Okay. And the R group is going to be on what's called the alpha carbon. So these are going to be talking about alpha amino acids. The alpha carbon just means it's one carbon away from the carbonyl carbon. Right? And so based on these R groups, they can either be hydrophobic or hydrophilic, right? And if they're hydrophobic, they're going to turn away from that watery environment that they're being synthesized in. If they're hydrophobic, they're going to be, revealing themselves. Depending on what those R groups have on them, they may form hydrogen bonds. Cysteine is an amino acid that has sulfur as an R group, and so we can form disulfide bonds. All of these things give proteins their structure. Okay? And proteins vary in structure based on the sequence, just like genes vary based on the sequence of A's, C's, G's, and T's. Just like computers, right? The codes are all just a sequence of zeros and ones. Right? It's binary code. DNA is quaternary code. Amino acids are 20. I don't know what the word for 20 is, but 20-nary code. All right? And so they come together and form this peptide bond here. Okay, the amine group of one acid comes here. And so you'll always have, at the start of the protein, you'll have what's called the N-terminal end, because it starts with the amine group. Okay? And then it chugs along in this order. So the back end, the engine, if you will, if we're thinking about these as trains, is the amine group. The caboose is going to be the carboxylic group. Okay? And then this is telling us here, right, that it's the R group is one carbon away, so it's called an alpha amino acid in that case. All right. Turns out that most amino acids are L amino acids. And that just means, remember we talked about carbohydrates, we said that most carbohydrates are D, right? And I said that we don't really know why. Just could be that the enzymes broke down a D conformer more efficiently. L-amino acids just refers to the amine group that is on that chiral carbon. And it's analogous to the sugars, right? So this hydroxyl group on the sugar is D. Here, well, in this case it's L, but most of the sugars are D. Most of the amino acids have this L. And they, again, it's unknown why that is the case. Okay. It has been shown that, believe it or not, a lot of meteorites have an abundance of L amino acids on them. And so one hypothesis is that the biomolecules on this planet were seeded from meteorites coming down. And so, if that were the case, then it's possible that it just happened to seed this planet with an abundance of L amino acids, and then that just became the bias. Right? But who knows, right? It's hard to say. That's just the hypothesis. So what else to say? So alanine, serine, those are amino acids that make up proteins. These are part of the 20 amino acids. But there are hundreds of amino acids out there, okay? And not, the vast majority of them don't make proteins. They have other functions, okay? So I believe I have a handout that has all of these amino acids on there. Like I said, they're broken up into 3 different categories: neutral amino acids, acidic amino acids, and basic amino acids. Okay. These are all, and then the next slide, are all, the majority of them are going to be neutral amino acids. And we'll get into what makes them neutral in just a second. But you don't need to know all of these. When I took a biochemistry class in college, we had to memorize all 20 amino acids and their structures and their abbreviations. And there's single letter abbreviations, and I thought that was it much when I was a student, so I'm not gonna do that to you folks. But for instance, what I want you to focus on is the blue and the pink are the common denominator, right? The amine group and the carboxyl group, the carboxylic acid group, and then what makes them different is this R group. So alanine is one of the simplest amino acids. Histidine is the simplest. Unless it's on. The R group is just a hydrogen. Okay. Then we have alanine. And then cysteine, like I mentioned, it has a sulfur there, right? So that can form disulfide bonds with another cysteine further down in the peptide. Okay. Oh wait, glycine has a hydrogen too. Isoleucine. And so in these names, you'll find similarities. So leucine is an amino acid. Isoleucine is an amino acid. And the main difference is just that R group. So if we look at leucine, the next one here. Yeah. So if we look at leucine, here's the alpha carbon, here's the amine group, right? Then we have 1, 2, 3, 4. And it's coming off in this way for leucine. But if we look at that for isoleucine, right down here, here's the alpha carbon, And we still have 1, 2, 3, 4, but it's just at a different carbon, different positional carbon. Okay. Alanine. So like I said, it's just a methyl group, but there should be a phenylalanine on there somewhere. What do you think a phenylalanine is going to be like? It's going to be like alanine with a phenyl group, right? So here's the alpha carbon, here's that hydrogen. Instead of a methyl group, we have a phenyl group coming off of the methyl group. Okay. So again, these are still neutral. Then we get into acidic. There are only a couple acidic amino acids, and what you'll notice is that the R group has a carboxylic acid group. Right? So that's why they're acidic, because in addition to the carboxylate ion on one end, they have a carboxylate ion at the other end as well, which is going to then drive more hydrogens into, the solution and make it somewhat acidic. And then the basic ones, well, what do we see on the basic ones?
Nitrogen.
An additional amine group, right? And so that is going to be proton accepting, right? And so it's going to create a situation that is more basic. And of these main 20 amino acids, we're able to make 11 of them. Okay, so we can synthesize 11 amino acids from their precursors to then utilize them. That means there are 9 that are called essential amino acids. And those are going to be things like lysine and valine. That's 2. What else? Leucine, methionine, phenylalanine, 5. Threonine, 6. Tryptophan, 7. Isoleucine, 8. Which one am I missing? Isoleucine. Well, anyway, that's you don't necessarily need to know that either. But just to give you context on some of these, there's no real. I'm not sure of a rhyme or reason to why some of these we can make and some of these we don't. So I'm not quite sure. There doesn't seem to be a whole lot of trend to that. 6, 7. Maybe histidine, I think, is probably 1, 2. All right. Questions so far? So there are other amino acids, right? And here are some examples of amino acids that don't make up proteins, but are really important physiologically. So GABA stands for gamma amino butyric acid. And you'll notice that the amine group is not on the alpha carbon. So it's not an alpha amino acid. Not all amino acids have to be alpha amino acids, just the ones that we use to make proteins. GABA is a neurotransmitter, and so it is responsible for giving us a calming effect, actually. And so as a result, I don't know if you've heard of the drug gabapentin or not, but it is used for epilepsy treatment. It's used for anxiety, sleeping problems, nicotine withdrawal, alcohol withdrawal, things that are going to make people anxious. And the mechanism of action is that it essentially modulates ion channels. So we have these calcium ion channels that open and close, and when they do, they send signals, and that helps with neurotransmitters, okay? The gabapentin is going to essentially decrease the activity of these ion channels. And when the body senses that the calcium ion channels are decreasing activity, it's going to upregulate the release of GABA amino acid. And a higher concentration of GABA is going to, chill us out a bit. And then thyroxine, I'm not as familiar with that. It's a hormone that's made in the thyroid. And so it's a really, really important hormone that functions in a lot of different things, but thyroid is involved with metabolism, so thyroxine is going to aid with metabolism, digestion, heart, muscle, brain function, lots of things, okay? But neither of these are used to make proteins, okay? This amino group is on the alpha carbon. So you can have alpha carbons that aren't used for proteins. You can have non-alpha amino acids that are also on there.
So these kinds of amino acids don't polymerize?
They don't polymerize.
Okay.
And it's, partly because if you had these amine groups and these really bulky R groups, if you look at that list, the R groups aren't that bulky, right? Phenylalanine has a phenyl group on there, right? But most of them are fairly small, right? Smaller than that thyroxine, for sure. And so it's going to be hard to make this nice puzzle fit together, right? If you have these really bulky groups, and then that's going to affect the folding, right? If they fit together in smaller packets, then you get a nice, tight, globular protein or fibrous protein, right? But if the amino acid is separated from the carboxylic acid by a long chain, or if there are really bulky groups coming off, it would make for a kind of a weird looking protein. Weird, proteins are kind of weird in general, but like weird for a weird looking protein.
Yeah.
All right, so properties. These amino acids can be acids or bases. So just like water is amphoteric, right? It can be H3O or H2O. Amino acids can also be amphoteric and can serve as acids depending on what the pH of the environment is and each amino acid's relative pKa. They can donate protons or accept protons, all right? And so what you'll see here is that each amino acid has a different pKa value for its carboxylic acid group, as well as its amine group, and in some cases, the side chains as well, okay? And so each amino acid is going to potentially behave differently at certain pH. And in the body, what we call physiological pH, that's going to be 7.4, okay? And so an amino acid will be an acid or a base, or it will have the hydrogen, or it won't have the hydrogens, depending on what the pH is, okay? So in our body, that's going to be 7.4, and that means that some of these will be protonated and some will not be. And all you gotta do is look at this list to see what the values are and know what the relationship is between pH and pKa to then predict whether or not we're going to have something here where that's a carboxylate ion, but here we don't have an additional hydrogen. Right? So this pKa1 is going to be, but depending on what, I don't know what the pH is, this is just a generic thing, but what they're saying is this pKa1 for this carboxylic acid group is such that it's going to kick off that hydrogen. But the pKa2 of the amine group also is such that it's going to kick off this. And so we only end up with a negative charge. Okay? In this case, we have a positive, negative, and that's a net neutral. And that's what we'll call a, I like this word, a Zwitter ion. Okay? Not a Twitter ion, a Zwitter ion. All right. So here we are, we have physiological pH, and what that means is it's going to drive the amino acid toward a carboxylate ion, meaning it's going to kick off that hydrogen most of the time. That's why the arrow, the equilibrium arrow, is much more in favor of this setup than this setup, right? And then same thing with our amine group. Physiological pH is going to tack on the amine group. And so, for instance, this alanine, you end up having a scenario where you lose the proton off of the hydroxyl group, and you gain a proton on the amine group. So you have a positive and negative charge. Overall, it's going to be neutral. And so the way to benchmark for this is that if the, what color is this going to be? If the pH is less than 2.36, which is directly linked to alanine at 2.34. If the pH is less than the 2.34, then the uncharged form predominates. Okay? If the pH is greater than 2.34, then it's going to be the carboxylate anion. And that's for the carboxylic acid group. So then for the amine group, if the pH is higher than 9.69, we end up with, again, the uncharged form. If the pH is less than 9.69, we're going to get the cationic ammonium. Right? So, less than 2.34, no ion. Greater than 9.69, no ion. Alright? And so that's where we get these Zwitter ions. A Zwitter ion means that it has positive and negative charge. Okay? And if you put that in an acidic solution or a basic solution, it's going to give you a different product. Right? we take the Zwitter ion, here, positive, negative, we put it in an acidic group, it's going to protonate that carboxylate ion, right? Because this is more acidic than that, so it's going to turn this into a base. Even though carboxylic acid is an acid group, it becomes a base based on Lewis base, Bronsted-Lowry. It accepts the proton. Conversely, if we're in a basic solution, here, it's going to be the acid. Right? And what's going to accept a hydrogen? Yes, this is going to kick off a hydrogen and we're going to make water. Right? So that's what the Zwitter ion means. It means that depending on the solution, it can be positively charged or negatively charged. If it's neutral, then it's going to be, if the solution is neutral, it's going to be a net neutral. There's going to be charge separation, but the overall charge is 0. Right? And amino acids, based on this, are a lot like salts. So what is a salt? It's an ionic compound, right? It has a positive and a negative. It has a high melting point. It's soluble. Salts are soluble in water. Amino acids are that way as well. They don't really conduct electricity. They're not electrolytes like salt per se. Although I haven't tried to pass current through a branched chain amino acid solution, but they do have some similar salt-like properties. Here is the amino acid, again, alanine with no charge, right? So we have this is how it's often written generically. Once we have it at physiological pH, that's going to change things, right? Predominantly, it's not going to exist like this. It's going to have some kind of charge separation. Okay? It might just have one charge. It might have both charges, but typically, you're going to pull off this hydrogen here and put one on to the amine group, and it's going to look like this. Okay? So that's what the Zwitter ion is. This is the Zwitter ion. This is just a generic uncharged amino acid. Okay. Questions so far on this? Pretty straightforward. All right, so as I mentioned, amino acids are amphoteric, meaning they can be positive or negative, which makes them acidic or basic, right? So in that first reaction here, we're treating the amino acid with a base, right? So if we're treating it with a base, what is it doing? It's taking a hydrogen off. So what is that amino acid serving as? The conjugate acid or base? We're treating it with base.
Conjugate's base because it's missing.
This becomes the conjugate base, right? Here, it's donating, or it's getting rid of a proton. So this is going to be, in this scenario, when we put this amino acid in a basic solution, it's going to be an acid. Okay? And then conjugate base, this is the conjugate acid of this base. Down here, though, we have it in an acidic solution, so then the amino acid is going to be a base. It's going to accept the proton that this donates, right? Right there. So just like water is amphoteric, can be an acid or a base, amino acids can be acids or bases depending on the pH of the solution they're in. Okay? And physiological pH is 7.4. So not quite neutral, it's slightly basic. So if it's slightly basic, the amino acids in our body exist in this kind of form. So physiologic amino acids will be written with that carboxylate ion. Okay. And then typically no charge on the amine group. Okay, so let's do a little practice here. This is lysine. What form is going to predominate at a pH of 9.5? So this is, you want to get out your charts here and figure out what to do with this one. So pH is greater than 2.34, right? So what's that going to do to the carboxylic acid group? And then the pH is just below that 9.6, so what is that going to do to the amine group? Let's see what you come up with. And then remember, this is for lysine. This was for alanine, right? So you got to look up the pKa values of lysine. All right, so what is the pKa for lysine?
2.18 for the carboxylic acid.
Sorry, what is 2.18?
Uh huh.
Okay, so 2.18 for the carboxylic acid. And what was the pH? 9.5. Okay. So, if the pH is less than the pKa, you're going to get the protonated form. If the pH is greater than pKa, you're going to get the unprotonated, the carboxylate ion. In this case, the pH is significantly higher, right? So we're going to get the carboxylate ion form. For the amine group, if the pH is less than the pKa, you get the protonated form, so the NH3+. If pKa is higher, or I'm sorry, if pH is higher than pKa, we get the uncharged, that just the NH2 off of that R group. So what is the pKa2 for the amine group?
It's 8.95.
8.95. All right. So pH is greater than pKa, so we're going to stay as it is. All right. So this would be a decent exam question. Right? I wouldn't make you memorize all the pKa values. I'd give you a table, right? And it would be up to you to be able to know that little deciphering tool right there.
So if the pH like was higher, or if it was dropped below that pKa for the nitrate group or whatever, would it affect both of them or just the one on the
No, it
alpha
just
carbon?
depends. So if we dropped it below 8.95, but it was still higher than 2.18,
Right.
it would just affect the amine group.
Okay. Would it just be the alpha amine group, or would it affect the other one on the side chain too?
Just the alpha.
Okay.
Want to practice some more? Let's try it. Or, well, actually, no. It would affect this. That's why these are all highlighted.
Okay.
Just like so with the acidic amino acids, it would also affect both of these groups.
Okay.
So, yes. Strike that previous thing. Yes, it affects all three.
Okay.
All right, so let's do a little practice, go over this, and then we'll take a break. So it gives you pH for each of these. You'll want to look up their pKa's and then ask yourself, is it greater or less than for each respective pKa? I'm going to fill up my water bottle.
Still going? Almost wrapping it up.
Yeah, last one.
Right.
Alright, so alanine, right? And it was pH 10. Alright. So for the carboxylic acid group, pH, in this case, pH is greater than pKa. And if that's the case, then we have an unprotonated ion, right? So it's going to look like that. What's the pKa? 9.69. So in that case, pH is greater than pKa, right? And when that's the case, we have the uncharged, so just NH2. So this is going to be NH2. Okay, everybody get that? Nice. And yes, I'm being lazy. I don't want to draw out this kind of since I've already done that. All right, what's the next one?
Proline at pH 10.
10 for proline also. Okay. So proline, pKa for the carboxylic acid is 1.99. So pH is greater than pKa, and pH is less than pKa, right? Okay, so when it's greater, it's ion. When it's less than, for the amino, it's protonated, right? So it is this form here, yeah. This is protonated. You can't, doesn't really show up well here, but that's a plus sign right there because it has 4 bonds. So it would look like this. Everybody get that? All right. What's the next one?
Tyrosine at pH 9.
Tyrosine at 9. Must be, is it the very bottom there? Yes, it is.
Oh,
Okay, so pH 9, that means our pKa is less than. What is less than?
Deprotonated.
Deprotonated. If pH, well, pH is less than, so it's going to be protonated, right?
Okay.
Because our pH, you said, is 9.
Oh, yeah.
Okay, so this is going to have an H on there. Oh, look at that. Perfect. Alive. What's so fun? You don't like that H? I just feel like this is the same color. All right, and then PK. Oh, I wrote on the wrong one. Shoot. All right, there. Now, pH is less, or pKa is less than. So pH is less than the pKa. So for the amino group, it's going to be protonated. So it's going to look like this. It's going to stay there. Okay. So this would be charged, right? In this case, it would be a +1 charge. Okay. Does that make sense? What else did we have? What was the next one?
Asper, I don't know. I don't know how to say that one. Asparaginine?
Aspartic acid?
Yeah. Or no, asparagine?
Oh, asparagine.
Asparagine.
Asparagine. Don't make any asparagine statements for asparagine. Asparagine. So, what's the pH for that?
Physiological.
Okay, 7.4. Asparagine, 2.02. So pH is less than pKa, so it's going to be protonated. So asparagine is protonated on the carboxylic acid group. Right? pKa is higher, so pH is less than pKa. Oh, still less than, right?
Yeah.
Did I make a mistake? So in this case, pH is greater.
Yeah.
pH is greater than pKa.
Yeah.
Sorry.
Yeah.
So then, yeah,
That's what I was trying to get at.
I think I just flipped those. If you flip it, it's going to be the opposite, right? So make sure you don't, if you want to have pKa first or pH first, it doesn't matter. Just make sure it's consistent. Don't do what I did. Okay, so yes, pH is greater than pKa, and so it's going to be unprotonated. It's going to be like this. In this case, pH is less than pKa, right? Because it's 8.80. And when it's less than, it's protonated. So it's going to look like this. And that should make sense because I said at physiologic pH, that's how these are written. It's a zwitterion at physiological pH. And that's one of the reasons that our body is a very stable 7.4. Because if you start tweaking that, then we'll get charges on these amino acids. Okay. It's not the reason that we have a stable pH, but the pH needs to be homeostatic in part so that we don't start getting charges on our amino acids. That would lead to different folding properties. And you probably know that the shape confers the function of a protein, right? If you change the shape, you change its function. What else? Is that it? Do we have more?
There's histidine at physiological.
Okay. Histidine at physiological. So, 7.4 pH is greater than pKa, and pH is less than pKa for the amine group. Right? So pH greater, it's going to be unprotonated. And that's what that is, right? The histidine. pH less than, it's going to be protonated. So it's going to be like that. So kind of, you can kind of cut all those comparisons out. If you ever see physiological pH, it's going to be unprotonated and it's going to be protonated.
It's like automatically zwitterion.
It's going to be a zwitterion.
Okay.
Yeah. Yeah. All right. That's probably why there are two of them in there, just to show you that. Yes, you can change the amino acid, but if the pH is still at 7.4, you get the zwitterion. All right. What? I should have done it this way all along. Blue. Okay. Then we'll say pH. All right. What's the next one?
Glutamic acid.
Okay. And what's the pH?
3.
3. Okay. So for glutamic acid, it's going to be pH is greater than pKa, and pH is less than pKa. Right? Okay. So if pH is greater, we have the unprotonated ion. And so this would actually be called glutamate instead of glutamic acid. This would be aspartate. So sometimes with these acidic ones, when we deprotonate them, you'll hear them called the 8 instead of the ic acid. And then pH is less than pKa, so protonated. Protonated. Okay. Is that it?
Yep.
Okay.
Would the side chain be proteinated or protonated?
Yes, so that goes back to that question. So this side chain would also, whatever this is, this will also be.
Okay, then how do you use the pKa of the side chain?
It's going to be the same.
But the values are different.
The
That's
chart
what I'm
has a
confused
different
about.
one, yeah.
Oh, this side chain here.
Yeah.
Yeah, so you would say 4.25 is still. Oh, I see what you're saying. Okay, I got you. Yeah, okay. So pH is 3, and the side chain is 4.25. So pH is less than. So in that case, it would be protonated. Most of these sites, so most of them don't have values. It's only the acidic and the basic ones. I think there might be, yeah, so cysteine, because it has a sulfur, that sulfur is going to contribute to its own pKa value. But yeah, good catch there. In which case, wasn't histidine one too? So we should have looked at that one as well. So if this, what was the pH for histidine?
Physiological 7.4.
7.4. Okay. So pH is greater than the pKa of the side chain. So uncharged. So it would exist like that, which is how they have it written. All right, any other questions? All right, let's take a break. Let's take a